this lecture, we are going to talk about processing, structure and charging of transfer RNA. Main points of the lesson are processing of transfer RNA, structure of transfer RNA and transfer RNA charging reaction. In a typical mammalian cell, approximately 80% of total RNA is ribosomal RNA, 15% is transfer RNA and protein coding messenger RNA constitute very small portion, that is 5% RNA. What is processing of transfer RNA? Mature cytosolic transfer RNA consists of average 75 to 80 nucleotides. They are produced from larger precursors, that is pre-tRNA. And pre-tRNA is synthesized by RNA polymerase III enzyme inside the nucleus in nucleoplasm. The precursor of tRNA is much longer and it consists of up to 350 nucleotides. So, pre-tRNA undergoes extensive processing. Processing of pre-tRNA like mRNA processing that takes place in the nucleoplasm. And processing discards useless sequences. This is done by endonuclease enzymes. Nucleotides are removed from both ends, that is 5' end and 3' ends. Some pre-tRNA they contain one or more introns that are spliced out during processing. So, endonucleases enzymes also remove many sequences and cleaving is done after the primary transcript has folded and formed characteristic stems and loop structure by extensive complementary base pairing. The mature tRNA is then transported from nucleus to cytoplasm through nuclear pores, and transfer RNA are generally associated with amino acids, that is proteins, and spend little time free in the cell. Now, structure of transfer RNA. Transfer RNA is a bifunctional molecule that must be able to interact with amino acid and messenger RNA. The structure of tRNA is highly conserved in the living systems and it can be formed into a clover leaf structure based on intramolecular base pairing that produces double stranded regions. The primary structure is then folded into L-shaped molecule that has two functional ends, the acceptor stem and anticodon loop. The acceptor stem is 3' end of the molecule and amino acid is attached to this end of molecule. So, base pairing within the molecule that creates three stem and loop structures in a characteristic clover leaf shape. The loop at the bottom of the clover leaf contains anticodon sequence which can base pair with codons of messenger RNA. The anticodon arm contains anticodon. It is a sequence of 3 nucleotide bases, and these 3 nucleotide bases they are complementary to the genetic code of messenger RNA for that particular amino acid. So, the two RNAs, that is transfer RNA and messenger RNA, they are anti-parallel and each mRNA is directed from 5' to 3'. So, this is messenger RNA and messenger RNA is directed from 5' to 3' and this is transfer RNA and transfer RNA is also directed from 5' to 3'. But these two RNAs like tRNA and mRNA they are anti-parallel to each other. What is a charging reaction? Enzymes like aminoacyl-tRNA synthetase must be able to recognize a specific tRNA molecules as well as their corresponding amino acids. The reaction catalyzed by the enzymes is called charging reaction and the product is an amino acid is attached to the 3' end of tRNA molecule. Now, this tRNA is called charged tRNA. ATP molecule provides energy for this endergonic reaction and the charged tRNA produced by the reaction is an activated intermediate that can undergo peptide bond forming reaction without an additional input of energy. As we know that amino acids are organic compounds that contain amino group and carboxyl functional groups along with side chain R which is specific to each amino acid. So the charging reaction joins the acceptor stem, that is 3' end of tRNA to the carboxyl end of an amino acid. A peptide bond is a chemical bond which is formed between two amino acid molecules when carboxyl group of one amino acid reacts with amino group of second amino acid with the release of water molecule. This is dehydration synthesis reaction, that is also known as condensation reaction, and usually occurs between amino acids. So the correct attachment of amino acid to tRNA is important because the ribosome does not verify this attachment. Ribosomes can only ensure that codon-anticodon pairing is correct. In a very real sense, the charging reaction is actual translation step. To sum up, pre-tRNA, it consists of 350 nucleotides, and processing discards most of useless nucleotides. So mature tRNA, it consists of 75 to 80 nucleotides. There are 20 different aminoacyl-tRNA synthetase enzymes, each specific for one amino acid. So the enzymes must recognize and bind amino acid to tRNA molecule with anticodon that specify the amino acid. So the reaction uses ATP and produces an activated intermediate that will not require further energy for peptide bond formation. So transfer RNA that has two functional regions, one that bonds with the amino acid and the other that can base pair with the codons of messenger RNA. So the tRNA charging reactions joins the carboxyl end of amino acid to 3' end of tRNA molecule, and without charged tRNA translation cannot take place. So this reaction is catalyzed by 20 different aminoacyl-tRNA synthetases, one for each amino acid.